Greetings. My name is Ian Taylor, and I'm Director of Product Innovation here at BD Biosciences Informatics. And today I'm going to be talking to you about some plugins that we've made available for free in FlowJo Exchange for use in both FlowJo and SeqGeek, SeqGeek being our product for single-cell RNA sequencing data analysis. Just as I get started here, I want to point out that there is a chat window and also a Q&A panel available in the Zoom app here that you've all joined. And so if you have any questions as I'm going through either the live portion of the demo or for the actual slide deck, feel free to drop those questions in and I'll try to answer them as they come up. I may get a little bit behind as I'll be clicking through the slides and performing some analysis while you guys pop those questions in. We've got a pretty good group of people joining today, so I want to thank you all for taking the time. And without further ado, I'm going to jump into the slide deck. Again, just remember to stick around after the presentation. I'm going to actually try and analyze some data live using many of the plugins that we're going to discuss within FlowJo right after this little update on the plugins that we have out currently and some of the newer features that are available there. Okay, so a little bit of what's new in the FlowJo Exchange in terms of plugins. First thing I want to mention is we have this TriMap dimensionality reduction, which is the newest form of dimensionality reduction available out there. And we packaged that algorithm up right away so that people could start applying it to their data. It's purported to do a better job of describing global structures in data sets than other dimensionality reduction techniques such as UMAP or T-SNE. ViolinBox plots is a plugin that we have available for creating violin plots and box and whisker plots, which has received a lot of attention lately. Been doing a lot of different updates on that plugin, and so folks should definitely give that a test if you're trying to create some nice visuals in FlowJo. Going to SeqGeek just for a moment, we've updated the Seurat plugin quite a bit, and we're now using the latest Seurat package for single-cell RNA sequencing analysis in that pipeline, very popular pipeline from the Satija lab. We also have a iCellR plugin, which is useful for performing pipelines of analysis in FlowJo, but can also be used to do differential expression analysis batch-wise in SeqGeek. There's CytoNorm, which is a batch effect normalization algorithm created by the SACE lab. Same creators, the same people that created FlowSOM. And that's going to allow you to normalize out batch effects if you're doing longitudinal studies or things like that in FlowJo. We also have batch effect correction available for SeqGeek through the algorithm BatchLR, which is in a plugin we call BatchLR. And that's useful if you're combining data sets in sequencing. You want to be able to remove some of the technical variability that just occurs across different runs in that technology. We also have a really popular visualization tool called ClusterExplorer, which I'm going to try to illustrate in our demo after this slide deck. But we've made some good updates there that I want to talk about as well. And then we've just recently updated our CBA plugin to work with flex sets and two-dimensional clustering kits. So if you're doing CBA analysis in FlowJo, if you've got CBA kits that you've run to test the concentration of different analytes, you can now analyze that data directly within FlowJo, which is pretty cool. Okay, so first thing I mentioned I'm going to talk about is TriMap. But before I talk about TriMap, I want to talk a little bit about data structures. I know this is something that not a lot of biologists are super familiar with, so just to give you a little kind of rosy picture of how dimensionality reduction can work. The whole idea here is dimensionality reduction is useful for high parameter workflows. So if you've got 10, 20 different parameters, it can be really, really tedious to start looking through those parameters by hand. And so we want to apply some machine learning to them. But before you even do that, you've got raw data, and that raw data represents the maximum local structure that you could possibly get of the data. You're zoomed way, way in, and you're looking at single parameters or maybe two parameters at a time for features that you might find in a data set, such as T cells, B cells, so on. As you move out to more global structures, you might perform something like principal component analysis. And that's going to give you a kind of an overarching view of the data where you can see the major subpopulations popping out, but you won't be able to find very, very small subsets typically within a principal component analysis embedding. And so anywhere in between those two different milieus, you can find nonlinear dimensionality reduction. And this is going to include things that we know already, like T-SNE and UMAP. But we've now added a new dimensionality reduction embedding, which is called TriMap. And TriMap tries to do a better job of retaining that global structure that we get from principal component analysis, so that overarching relatedness between clusters within the embedded space. So how does TriMap look? How does it compare with T-SNE? Well, let's take a closer look. So to look at the T-SNE map compared to TriMap, you can see here I've just performed dimensionality reduction on the same data set 4 different times. If we start from the left, you'll see some raw data, and I don't have axis labels here, but we're looking at CD3 versus CD16. So we can start to pull out broad buckets of cells. Basically, we can see the NK cells pretty well, though they're mixed in there with the patrolling monocytes in the top left, and in the bottom right, we're gonna see our T cells. Everything else is just clumped into the corner in the double negative space in the bottom left. As we begin to apply some machine learning to that raw data, we can look at hundreds, as many parameters as you like. We can look at thousands of different parameters all at once in a T-SNE map, which is going to give us a better picture of kind of the local structure of the data. And we're going to be able to start to spot all the different subsets within those broader buckets that we can see in the raw data on the far left. Next up is UMAP, becoming more and more popular. At some point, we think probably more people will be doing UMAP than are currently using T-SNE, because it does a better job of retaining the global structure of the data set, meaning populations that are closer together in Euclidean space tend to be more closely related, whereas in T-SNE, that's not as much, that's not going to be guaranteed. So with that in mind, how can we maximize that? How can we retain the best global structure of the data? Well, mathematicians are hard at work on that problem right now, but the latest kind of preprint from the producers of TriMap gives us an idea how we could do that with a new algorithm. And so we've implemented that as a plugin, which you can now download from FlowJo Exchange, and we'll kind of show you that in a moment. And finally, principal component analysis. You'll see in that principal component space, there tends to be kind of 3 different populations that matches or mirrors a bit better the TriMap space. And these other nonlinear dimensionality reduction methods tend to miss that part of the broader structure of the data, if you will. Okay, so then we can also overlay subsets on top of that when we start to actually spot all those different populations I was mentioning. And you might notice that, let me grab my zoom controls here really quickly. I'd like to just be able to point to a couple of these different subsets. Not seeing it. Well, hopefully you guys can see my cursor all right. Basically, just to show that we have, say, B cells here. Those same B cell populations are kind of overlapping each other in the raw data space, but they're actually in dark purple and light purple. And if you see in T-SNE, that dark purple subsets are kind of separated from the light purple, whereas in UMAP, they're placed right together. And so this is what we mean by global structure of the data. We're actually seeing that there's an interrelatedness between these 2 populations within the UMAP space, which would be otherwise lost in T-SNE space. Similarly, in TriMap, you get an even better, we think, overview of that broader structure of the data by trying to retain this PCA-like structure to begin with. Okay, I think enough on TriMap. You guys can check that out for yourself. I'll probably actually run that algorithm when we get into the live demo a bit more. Okay, Violin and box plots. This is a visualization tool that we've developed for the purposes of creating some really, really nice violin plots and box and whisker plots. And so just to walk you through the panel here, we've got a section at the top that allows you to select different parameters. In this case, we're looking at parameters in gene sets because this is being run in SeatGeek, but the plugin will work in both FlowJo and SeatGeek. The important thing to note there is you've got to select a set of parameters at the top that you want to compare, and then a set of clusters on which you want that comparison made. So in this case, I've selected sample ID because I want to compare perhaps my different parameters there across a sample ID clustering parameter or categorical parameter. That's going to help us distinguish between the different samples on the basis of these few markers that I've selected. Then down below the parameter selectors, you've got a bunch of different options, little check boxes that you can tick, and just going to depend on what type of visualization you want to create. So in there, you can choose from violin plots, box and whisker plots, or jitter, or you can combine them in any way you see fit. Here, you can see they're all selected. You can also change the orientation of the figures that you'll produce, whether you want to flip those and make them kind of horizontal as opposed to having vertical plots, or you can overlay them, so you can group them together, which can be a nice presentation as well. You've also got options for different colors, dark theme, whether or not you want to create a PDF is an option. And you can add a stat table if you want to export statistics, just kind of as a summary from the analysis right away. And finally, you can create a heat map. And this heat map is kind of a nice feature. A lot of people request the ability to create a heat map of populations. And so this will allow you to do that within FlowJo. This is an example of some different outputs from that plugin, including box and whisker plots on which I've shown some jitter, and we've turned on the dark theme background, which I think would look really nice if your slide background was black, but here on the white background is probably not quite as nice. And then on the right, you see the heat map that's generated as part of that plugin as well. Question coming up, which parameters to choose within FlowJo, comp or non-comp? So I think we're referring back to this parameter selector window. You would choose the compensated parameters typically for any of these analyses, assuming that you're doing compensation, and you probably should be doing compensation. There might be some exceptions to that, like you may not be compensating your live dead or some dump channels, and if you're interested in looking at the expression of those markers, you can always choose the non-comped channels, but assuming that you want to see the compensation applied to the data, you'll definitely want to choose the compensated parameters. Great question there. Thanks for that. Like I say, do feel free to drop questions in on me as they occur to you. Okay, next up is a plugin that's very specific to SeatGeek, so I'm not going to kind of belabor the point here, but for many people that are doing single-cell RNA sequencing data analysis, they'll want to apply this very popular sequencing pipeline to that data, which is the Seurat plugin. The Seurat plugin can do things like dimensionality reduction, clustering, cell annotation, all kind of automatically. And we're looking at more ways to do this for FlowJo in the future. But for now, this is only available for SeatGeek, and it's going to work really well to start to kind of analyze and dig into your single-cell sequencing data if you guys are doing any of that currently. The type of graphics that Seurat is going to output, again, is going to include clustering. So on the left, we see violin plots associated with a set of clusters that were predicted by that algorithm. And in the middle, you can see some dimensionality reduction that was performed, specifically UMAP, that shows the presence or absence of certain marker genes for a variety of different cell types. And then on the far right, you'll see that we're actually printing out a heat map to show how those different cell types express or lack expression for certain markers. And this can be indicative of a cell type, which is sometimes very hard to figure out in this, ultra high parameter space from single cell sequencing. And these are other outputs that Seurat produces. You can see that all of those different dimensionality reduction parameters that are created, whether it be principal component analysis, T-SNE, UMAP, are all live within the SeatGeek workspace. You also get a set of differentially expressed genes for each of the parameters, for each of the populations, which will indicate sort of the profile of expression for that population, typically used to characterize these populations if you've got transcriptome information for them. Okay, next up is iCellR, which is actually a package of analysis tools developed by Reza out of NYU. He's a brilliant bioinformatician there. This does not incorporate every single feature from iCellR, but it's some of the really big highlights that we liked. And it can be run in both FlowJo and SeatGeek. Unfortunately, you won't be able to perform differential expression analysis in FlowJo. That's not really an analysis tool that people are using with Flow. But you can walk through the iCellR pipeline in FlowJo and generate dimensionality reduction and clustering. It's also got a kind of a cool feature that allows you to create 3D plots. And so sometimes I like to play around with this 3D plotting tool just to make funny kind of graphics. And so here you can see I've carved out a little message for you guys in CD4, CD45 RA space. And we've got a little bit of FoxP3 there on the Z-axis. But so you can create these 3D plots that are kind of interactive. And you can actually zoom in on your data, mouse over the dots to get some extra information, or look at clusters in a three-dimensional space. So that's kind of fun to do. We'll see if we have some time. I might be able to run this 3D plotting tool on some data we'll look at after the slides. Okay, next up is batch effect correction for flow data. This is the CytoNorm algorithm. I'm just showing you some different examples, both from the paper and also some that I ran in FlowJo. And the whole idea here is that as you collect data across maybe weeks or months or even years, your data will drift somewhat just due to the technical variability that you experience in the lab. This could be down to different technicians dealing with the data. This could be down to a whole different flow cytometer being used to collect the data, or just differences that occur across time, like PMT, drift across the PMTs. So in order to run CytoNorm, I just want to give a little cautionary tip here. You want to have the same controls run for every single sample. So some have chosen to freeze down some like PBMCs, maybe activated PBMCs, before they start their longitudinal study, and that way they can add aliquots of the exact same PBMC sample to every single one of their runs. And then that can be used as a control downstream with CytoNorm to try and remove some of those batch effects. So the idea is that you would have a set of parameters that you run with a normal analysis, and then the next time you bring in some new samples, you could actually normalize those samples to your old batch of samples or your initial batch of samples and continue your analysis from there. And this is showing some more images from the paper, just kind of showing that CytoNorm indeed does a better job of removing those batch effects than some of the other options that have been available traditionally. All of these are mostly going to be run in R, and so you wouldn't have access to all of the different options in FlowJo, but CytoNorm is now happily a plugin that you can run, so you can try to normalize out those batch effects if you want to. BatchLR, again, is a sequencing batch effect correction algorithm. It's actually also known as BatchLR. It's a mutual nearest neighbors type of correction that tries to align cells based on their set of mutual nearest neighbors in terms of high parameter space. And then we can kind of infer that these cells are similar to one another. Here are some results that I got from comparing just two very different PBMC samples, where you can see that initially uncorrected data in T-SNE space just separated out the different samples entirely. But because these are just healthy human PBMCs, we expect them to sit very close to each other, if not kind of right on top of each other. And so in the lower row, you can see that after correction, those samples sit right on top of each other just like we'd like. And of course, looks a little bit nicer if you annotate these different graphs. And so here you can see them annotated, and you might notice that, like, for example, in the B cells, we have one contiguous population here when we cluster, but if we look back at the uncorrected data, those same B cells are just separating out into clusters based on their sample compartment, which is not what we want. So this batch effect correction worked really well in this case. And just to kind of go back and forth, each of these populations separates out a little bit. I think this is classical monocytes, and you can see that it's basically the job of batch effect correction is being done really, really nicely by the BatchLR algorithm in this case. Okay, next plugin I want to show you guys during the demo is going to be ClusterExplorer. And so ClusterExplorer is just a really convenient way of looking at many different clusters across a high parameter data set. And here you can see I'm just showing a few parameters of interest, but we can show UMAP, TriMap, T-SNE space with those same clusters highlighted, and we can see their relative expression. This can help us to characterize populations of interest from clustering, or even to identify phenotypes that might otherwise be kind of hidden just underneath the generic cluster labels that are given in FlowJo. And then last but not least, we've just done a big update for the CBA plugin, which is a cytometric bead array tool that can be used to analyze CBA data. So actually a lot of researchers out there doing CBA, and currently they're forced to use other third-party softwares that are maybe not so easy to use. And we've tried to make this very, very simple, but also powerful. And it works in conjunction with all the tools available within FlowJo. So running CBA analysis with the CBA plugin, we hope would allow people to have a little bit more robust experience and be able to keep all of their analysis right there within the FlowJo ecosystem. I think with that, I want to just pause for a moment. Thank you guys for listening to my little spiel there on the new plugins and the updates. I'm about to show you some live demos of FlowJo in action, but I just want to make sure there aren't any questions that I've missed. Oh, it looks like there was a hand raised. Dan, if you still have a question, can you go ahead and just type that into the Q&A box? There should be a Q&A box associated with your Zoom app there, and I can kind of answer your question that way. Probably typing a question right now. Any other questions? Oh, comments on Phenograph. Dan. Oh, yeah, good point. So I want to show you guys Phenograph in the live demo. Phenograph is a really, really handy plugin. It's probably my favorite type of unsupervised clustering that's out there. Specifically, it's a Louvain style clustering algorithm. And we've done some good updates on Phenograph, things like it will now automatically install the packages in R that it's dependent on. And it's also going to delete its own plugin node and pop open a little graph window when it's finished calculating, so you'll know that it's completed. Yeah, really, really good question there, Dan. Thanks for asking that. And I will, like I say, try to show that as we get into the live demo. Okay, another question coming in. Examples and quizzes to practice. We don't have quizzes, no, but we do have demo data that's available on flowjo.com. I can show that in a moment. So yes, there is absolutely the option to use demo data. Question about T-SNE. When dots are shown close together, the question is, what does that mean when those, I think the question is, what does it mean when dots are close together in T-SNE? Well, if they're part of the same exact island, I'll say that they are probably the same cell type, or at least fit into a broad category that's similar to their closer nearest neighbors. But if they're in totally separate islands that are kind of distinct in Euclidean space, that doesn't necessarily mean that those two islands are not closely related to one another. Sometimes similar islands are just pushed apart because of the differences that occur there, but maybe they're actually more similar than islands that occur between the two. So that goes back to that whole global, local structure of the data that I was talking about. If you want to kind of ascribe a more biological meaning to those islands or to the clusters that you see in dimensionality reduction space, I would strongly recommend using something like TriMap and see if that does a better job of kind of mapping the location in terms of like phenotypical space for your data set. Hopefully that gets to that question, but feel free to ask me again because or rephrase, because that's a really important question. Question about a heat map with different antibodies and different MFIs. Can we normalize these using a z-score? We don't currently have any heat maps in FlowJo that utilize a z-score statistic, but that's a great idea. Currently, we utilize a normalization based on mode within the ClusterExplorer tool. And so I can kind of show you that as we get to the end of my live demo. So definitely see a heat map with some normalization, but not a z-score. No, that's a really good idea for future work. And question about, so similarly in single cells, question about dimensionality reduction when dots are close together, again, are the populations assumed to be related within single-cell RNA-seq? And I would say yes. In any type of data analysis you're performing, whether it's, it doesn't even have to be for biology, right? It could be for any kind of, like even demographic research, they'll sometimes utilize some of these dimensionality reduction tools. And if you have populations that sit very, very close together in those embedded spaces, then it's sort of assumed that those things are closely related. Of course, it's always advisable to go do some other work to kind of validate those findings and just double check that the that the factors that make those things close together in high dimensional or in embedded space are also present in the higher dimensional data. And yes, this this whole session is being recorded, and so it will be published after we finished with the webinar. Okay, and I'm going to go ahead and close out of the slideshow. I'll leave this open here in case I need to refer back to any of those figures. But I want to start digging into an analysis right within FlowJo. Before I do that, I want to talk a little bit about the setup of plugins. So if you guys are using plugins, and obviously anyone who's joined this webinar is probably interested in plugins, you'll notice that on the FlowJo documentation site, docs.flowjo.com/D2, we have an entire chapter, an entire section devoted just to plugins. And probably the most important out of all of those pages is just the setup of plugins. So installing plugins is a web page that I think anyone who's interested in plugins should definitely check out. It's going to talk a little bit about how you set up plugins going forward. There is a little bit of setup involved. It could include simply installing the plugin itself, the jar file for the plugin, into your plugins folder, or it could be that you're using a plugin that depends on R, and so you have to actually generate a, you have to actually add the plugin package to the R environment in order to utilize it. For an R version, we definitely recommend using the latest. So this would be R version 3.6.2 currently. And we try to keep up to date and make sure that all of the plugins work with the latest version of R. Whenever they release a new version, we always have to double check that, because since R is a third-party software, they could change things and break some of the plugins. But generally speaking, yeah, we're able to use the latest version of R. So R version 3.6.1 or R version 3.6.2 would be preferred. In terms of dimensionality reduction, another question came up from Dan just asking which is the preferred dimensionality reduction for high dimensional data or high content data. And I definitely recommend trying the TriMap option out, because that is kind of the latest and greatest. But you should use your own kind of best judgment. And if you notice that TriMap isn't doing the job you expect it to, maybe try T-SNE, because obviously that's going to give you a totally different representation of the data based on the local structure of that data. Okay, so the setup of plugins, again, it's going to involve obviously having FlowJo installed. Once FlowJo is installed, you'll notice you get a plugins folder, or if you don't find a plugins folder in some of the newer versions of FlowJo, we're not including that. You can create a plugins folder anywhere on your computer that you like, and just make sure that you add your plugins, the .jar files, to that plugin repo. At that point, when you restart FlowJo, you should start to see all of those plugins that you've added are going to populate on a list under the Workspace tab of the workspace called plugins. And in there, you'll see each of your plugins listed. One thing people will oftentimes note is that these plugins are grayed out when they first open up this plugins dropdown. That's okay. That just means that you haven't selected a population on which to run the plugin. So make sure if you want to run a plugin that you've selected a population, and that's just telling FlowJo where you want to run the plugin. Next, you're going to want to open up your preferences and make sure your preferences are all set okay, and I'll kind of show you that in a moment. If you ever want to get a new plugin, if you notice that you don't have a plugin that you know is out there and you're interested in using it, or you just want to know what's new, just check out FlowJo Exchange. This is the website, flowjo.com/exchange, where we actually host all of these tools. They're free to use if you are already have a FlowJo subscription, so we definitely recommend check that out and make use of these tools because they are available to you. And there's a whole suite of different features here. If you need to filter this, like maybe you're only using FlowJo, you don't want to use SeatGeek, you can uncheck SeatGeek, and now you'll only see plugins that can be used with FlowJo. So that's kind of helpful. And then if you're only interested in maybe seeing what's out there for dimensionality reduction, you can filter for that as well. Just going to turn off all these other options, and now we've just got anything that's going to utilize dimensionality reduction is all going to filtered for you that way. So question about FlowClean. FlowClean was not well supported, and so eventually I think we removed it from the site, but that's okay. We still have FlowAI, which is kind of a handy tool if you want to QC your data via, applying some machine learning to remove outlier events. FlowAI is probably recommended over FlowClean, which is just a little bit older and maybe slightly outdated. Okay, now you can see I've already got some analysis set up here. I want to just double check that my plugins are set very nicely in the preferences. So in preferences, check your diagnostics section, and you want to make sure that you have an R path set there. If you have no R path set, you're not going to be able to use R-based plugins. So let me make sure that I set my R path here. If you're on a Mac, there's a real easy way to find your R path, and that's to open up a terminal window. You just open your Spotlight, type in terminal, and then hit Command N. It'll open up a new terminal, and I can type in which R, and that gives me the R path. So this is how I like to find the R path. You can always try the default, which is this path here, user local bin R on a Mac. On Windows, it's going to be a little bit more complicated, but if you click choose here and go to your R application in the program files folder, it's probably going to be something like C:\/ program files /R / whatever R version you're using. So again, if you're on the latest, it's going to be like R-3.6.1. And then you need to go to the bin folder inside that folder, x64. And that's the default location for the R path on Windows. But of course, we're on a Mac here. After you set this R path, you have to make sure to save your preferences and then restart FlowJo. So I'm about to restart FlowJo just to make these preferences take effect. Also notice I've got a plugins folder in my applications, and that is correct. That is where my plugins are. You can see it's plugins under applications. That's where I've got all the different plugin jar files loaded there and ready for us to test out. But first things first, let's save this workspace. And I'll just save it into my downloads folder. We'll call it, I'll be a little bit suggestive here and say awesome plugin demo, and you'll all agree that that's accurate, right? With my initials, I like to revision my workspaces, so I'll call this revision 0. I'm going to save that, and then to quit out of FlowJo, the quick way is just to hit Command Q or Control Q if you're on Windows. And I don't need to save this because I just did save it, so no, don't save it. And then I'm going to go ahead and restart FlowJo. So I've got FlowJo here on my dock. If I just click on that, it should open up FlowJo. And while FlowJo is opening, I notice there's another question here around R. So if you've got an older R version and you update R, will you need to recalculate the data? No. So the reason you don't need to recalculate your data typically with a new version of R is because the calculations that FlowJo performs are all performed on the raw data using R, but then exported so that they're saved in like a CSV file in your plugin derivatives folder. So that's a great question. You definitely should not need to, let's see if this will work. You definitely should not need to rerun calculations just because you upgraded your R. Okay, awesome plugins demo, that's the one we just saved. I'm going to open that up. And then in this workspace, I'm going to go ahead and select just the singlets population that I want to run my plugins on. But because this is a demo and I want to give you guys all the kind of pro tips that we can offer, I strongly recommend running these plugins on a concatenated data file from all of the samples in most cases. So because that is the recommended workflow, I'm going to go ahead and try and follow it now. And the way I'll select all these singlets populations very quickly is just to go to the Edit tab here and choose Select Equivalent Nodes. Once I've got those equivalent nodes selected, I can just right-click on any one of them and choose Export Concatenate, and that will bring me to the Export Concatenate window. And the reason this is nice is because concatenation can take all 7 of these samples and turn them into one data file, which makes the analysis going forward very efficient and uniform. So it's very reproducible. It's perfectly reproduced across all the different samples. I'm again going to label this concat plugin demo. I like to add some extra keywords here, specifically $FIL can be a really handy keyword to parameterize. So anytime you have keywords that you don't want to lose when you perform your concatenation, you can add them as choices here under additional parameters. This is what we will sometimes refer to as a parameterized keyword. And I always make sure to uncheck this box for spreading the distribution of that keyword. Okay, once we feel we're set on that, I'm going to export this to my downloads folder. Then we'll click concatenate. And I want to load this into the existing workspace so we can continue on from the same spot. And I'll click close. That'll load the data into the workspace and then cancel out of this export concatenate window. And now I've got this new concatenated data file, which I can use for the rest of this analysis. I'm actually going to put that into its own group, which I'll call concatenation. By just dragging that sample from the all samples group into the concatenation group. Okay. And now these gates are somewhat meaningless. I'm just going to kind of minimize them here. And I'm going to begin by running some dimensionality reduction. And so again, the question was, one of the questions was, what's my favorite dimensionality reduction? Well, obviously it's TriMap. It's the latest and greatest. And so I'm going to go to the workspace tab here and visit the plugins section where you can see I've got a long list of plugins, and that's just because I've got all the different plugins available installed there, but you probably won't need to have all the plugins. You just want to pick and choose those ones that are most useful for your analysis. And then within TriMap, I want to make sure that my data is compensated. It looks like I didn't compensate that file, but there's an easy way to do that. All of these samples share the same acquisition defined compensation matrix. And so I can drag and drop this M icon onto the concatenated data file, and you'll see there's a little compensation badge that shows up there. Now my concatenation is also compensated. After having selected my compensation, or my compensated concatenated data file, now I'm ready to run TriMap. So back to the plugins, you can see again, all the different plugins that we have available are showing up on my list, but I really just want to run TriMap. So I'll select that plugin, and then that's going to pop open this dialog where I can select the parameters that I want to utilize for this dimensionality reduction. And because this is a very robust sample, I think it's got 18 different colors in it, I'm actually going to utilize all of those parameters with the exception of my dump channels. And that's going to include this guy. Oh, I'm sorry, that's not dump. It's just the okay. Yeah, I'm going to actually, it's going to be all the parameters because I haven't done any gating on the fluorescent channels. I want to keep all of this information in there when I run the TriMap plugin. Different distance functions that are available for this algorithm are Euclidean, Manhattan, Angular, Hamming. I have found that none of the changes here to the distance function makes a huge difference for the results. Otherwise, I would typically choose Angular here. You can choose the number of nearest neighbors, which is going to help to pull populations together or push them apart. So recommend, you could adjust these settings, but the defaults work pretty well. So I'm going to test with the default now. And when I click OK, it's going to begin to calculate that TriMap dimensionality reduction space. TriMap is actually coded in Python, and so it has no extra dependencies other than the PyInstaller that comes inside the plugin. So you shouldn't have to install Python or install R or any other setup to make TriMap work. It does come with one caveat, and that's that TriMap will only work on Windows 10 or Mac operating system. So if you happen to be using Windows 7, you're probably going to have to upgrade soon anyway, and at that point you can start using things like TriMap and UMAP, but until you do, you won't be able to use either of those Python-based plugins in FlowJo. And we kind of just wait for this thing to calculate. While we're waiting, I could actually start running Phenograph. That's going to be the next plugin that I want to run. So I'm going to go ahead and select Phenograph. And again, in Phenograph, we're going to select all the parameters of interest, which are typically going to be the same set of parameters that we use with TriMap. Now, because I've got 50,000 events, I think K of 30 is going to be a little bit low. Let me set that just to 50. I might go as high as 100 for this data set, but the higher your K number, the longer it will take to calculate. And I want this to happen very quickly, so I'm not going to go any higher than 50. I also like to change the run ID here, but if I change the run ID, it's going to have downstream effects on ClusterExplorer that I don't like. So if you're going to use ClusterExplorer, recommend you just use the default or auto run ID for Phenograph. And that goes for any clustering that you're running. you should try to use the default if you're going to use ClusterExplorer as a downstream step to try and explore those clusters a little bit more. The reason I can run, by the way, the reason I can run Phenograph and TriMap simultaneously is because TriMap is running on a separate thread in Python, and Phenograph is running in R. So Phenograph does have some dependencies in R, and definitely want to make sure that you have all of those R-based dependencies set up for Phenograph. This includes the extra step of installing R tools on Windows or Xcorts on Mac. So you need either Xcorts or R tools present, depending on your OS, and then Phenograph should run quite well. And like I mentioned before, the clustering that Phenograph generates is actually quite robust, and so I like using Phenograph partly for that reason. Okay, TriMap finished calculating. And we know that because we got some derived parameters calculated in the workspace. It also automatically popped up this graph window and deleted the plugin node. When a plugin finishes calculating, you don't actually need that plugin node to stick around in most cases. And so you can go ahead and just delete that if it's kind of stuck in your workspace. But for the newer plugins, we're trying to make them automatically delete their plugin nodes whenever that's appropriate. So here's the TriMap dimensionality reduction that we are given back as a contour plot. And so oftentimes I'll want to go ahead and add this to the layout editor. Of course, we have some options to do that. I could just drag this plot right into the layout editor, or I could go up here to the edit tab and choose copy to layout editor. But you might notice in some of my demos, I typically just click Command L, and that should automatically add the plot to the layout. Oop. Sometimes it takes 2 tries. Let's try that one more time. There we go. And that's just a hotkey to get this plot into the layout. But if you notice me just popping plots into the layout editor willy-nilly and it looks like magic, it's because I'm using that hotkey. Question. Finding that T-SNE works very well on 20 to 50,000 events, but more than 100,000 events, it gets slow. And what sort of counts would we recommend on a normal computer? So first off, let's define what a normal computer is. A normal computer, our recommended system specs at least, is 16 gigabytes of RAM and a typical like i7 processor, nothing too fancy there. Mainly your bottleneck is usually going to be found in the amount of RAM that's available on the machine. From there, you'll probably want to select some of the more advanced options for T-SNE if you're doing more than 1 million cells, but greater than 100,000, it should be pretty fast. I would anticipate you calculate a T-SNE in, say, under 5 minutes in FlowJo, which is extremely fast compared with some of the other algorithms that can run T-SNE. This is, of course, not a plugin. T-SNE for the latest versions of FlowJo is not found in the plugins list anymore. Instead, you can run T-SNE natively within Java by clicking on this T-SNE option here. I'm not going to run T-SNE for this demo because that's covered in a lot of other webinars, and also it's kind of old hat at this point, and it's not a plugin. So, but yeah, so you can probably improve the performance for very large numbers of cells by choosing one of the different gradient descent algorithms there. That'd be my first suggestion for improving the performance of T-SNE if you've already kind of maxed out your system in terms of what you can add for RAM on the computer. Okay, and then Phenograph finished, and you'll notice again it kind of deleted the, yeah, no problem. So ran Phenograph in no time flat, and we get a bunch of different clusters output. I'm actually not sure what the number is here, but it looks like quite a few. So we got 31 clusters predicted by Phenograph within this sample, and that's a bit more than I expected, but that's okay. We can definitely continue on with those clusters. Basically, the next step is going to be to start to explore what these clusters are doing within the data matrix. And so for that, what I'll often do is just duplicate this TriMap plot and then overlay my Phenograph clusters on top of TriMap so we can see how they distinguish in that embedded space. Currently, we're looking at a contour plot, so all of our overlays show up in contour. I think I would prefer to see those as dots. So let's switch to dot plot. And hopefully we'll see that these different populations kind of segregate out into different islands within the TriMap space. But you can see how it's potentially kind of indicating a relatedness between these clusters on the basis of their position within the embedded space. So that's kind of one of the really cool highlights for TriMap. You could go through and gate these cells to kind of get your major subsets and then start to figure out which populations might be of more interest to explore further. Or you could apply something that we're about to do, like ClusterExplorer, to try and understand what those phenotypes are within any given set of clusters. So without further ado, let's go ahead and run that. This is just, again, another plugin. So we'll go to the Plugins tab here and click on ClusterExplorer. ClusterExplorer is another plugin, by the way, that has no dependencies in R or other outside parties, other third-party softwares. So you can just run it by placing the jar into your plugins folder, and because it runs entirely in Java, it will just work for basically any machine. We can, of course, explore all the different parameters that we utilized for clustering, but let me select just about half of them because we've got, so many parameters here. I don't want to kind of overwhelm, but I do want CD19 and CD4, so the ones that I'll recognize very easily. Turn some of these off. Okay, and then this memory marker, I'll leave that in there. Okay, and when you're ready, once you've selected a categorical parameter, which defines your clusters, like Phenograph here does, and your parameters of interest, you'll also probably want to make sure that you have selected derived parameters like trimap X and Y, or T-SNE 1 and 2, or UMAP, whatever you want to look at this data on top of. And that's not required, but that's just a really nice to have. It's going to make for a really nice figure. Then you'll click OK, and it will begin to calculate the graphics that it wants to place into your ClusterExplorer window. So this is ClusterExplorer, and you can see inside of ClusterExplorer there's a few different panes, and I want to describe what each one of them does. So in the top left, we're going to see a pane that's what I call a chord plot, what's also been called, it's basing the other name people use, but cluster profile plot maybe is another word for that. This is just showing you the amount of expression for any given parameter across all of your clusters that you've discovered with Phenograph. And there's some more functionality in this plot that I'm not going to show right away because I want to kind of keep that overarching view of everything. But in the bottom left-hand corner, you see that heat map that I was referring to. So this is a heat map that's normalized to the mode expression for each of these populations. And you can actually scroll through each of these populations, and everything in this plot will update live. We'll see that in a minute. Bottom right-hand corner of the ClusterExplorer is that T-SNE plot that we selected, which shows in color, of course, beautiful color, similar to our overlay, each of the Phenograph clusters, and you can always change these colors as well. So now if I wanted to kind of scroll through these populations, or maybe some populations stand out as particularly interesting, let's see if we can find some T4 cells, for example. We can look for CD3 expression, and you can see all of these ones that are red are probably our T cells, right? Not difficult there. But now if we kind of cross-reference the T cells with CD4 expression, you'll see that there are some CD4 expressors that are not CD3 positive, and of course those are probably our classical monocytes that are popping up there. So this cluster here is very likely candidate for that. Looks like we don't actually have CD14 included, at least in the parameters I selected, but that's okay. Here we see a population that's very, very bright for CD4 expression, also CD3. We can probably assume that this is probably a T4 population. And now if I select it, it actually updates everything in the graph, everything in all the panels kind of live, if you will. And so we can see that this population is also highlighted. Now, as I scroll through, if I just select one of these clusters, I can actually scroll through each of the clusters and show how each of them change position within the trimap space, and also what their relative expression is. Here I found another one that looks like it might be a myeloid population out there. It's got CD4 expression, but no CD3 there. Okay, keep scrolling. There's one that's just negative for all. This one looks interesting. Again, CD4 bright, CD8 sort of bright, and it looks like a very, very small population. This is probably some double positives in there in the periphery. I believe this is PBMC data. Here again, you can see a population that's totally unrelated to the T cells, and it's appearing in a totally different location within the trimap space, and so on and so forth. We can kind of scroll through all of them. I didn't really notice where the T8 cell populations are sitting here. So let's look across CD8 and see, okay, this population here, for example, is probably cytotoxic T cells. And that means to me that probably all of these are in some way cytotoxic T cell like. The NK cells oftentimes get mixed in there as well. Looks like I don't have an NK marker here that I would recognize, so I'm not going to try and find that, but just to note that that's there. Then double clicking on any of these plots will bring back the total set of populations. I believe I can do that here or here. You can also change, let's see, you can also change the color palette here for the heat map. So if you right click, you can copy the text of the, copy to text means copy the MFIs for these different populations across the parameters selected. And then you can paste that into Excel if you want to get a table of those values. Or you can change the color palette. So under heat map palettes, I particularly like the green, black, red color palette. I think that's very stark and it looks pretty nice. You can also obviously right click and copy the image. If you're copying the image to paste this somewhere, I strongly recommend pasting it into PowerPoint. So I can kind of just paste the heat map in there if I like. And that will give me some information about each of the clusters across those parameters. I could do the same thing with the chord plots or the tri-map as well. Let's do that for tri-map now, just to show that. And I showed very quickly that if you double click on a cluster here, you can actually change the color. So if you don't like this brown color, I can change that to a nice foresty green color or something, and that will change here. And also it will change the color of the chord plot up above. The last thing for ClusterExplorer I want to illustrate is that you can actually do some gating directly on the chord plot. So let's pick one of the examples that I was using before. Let's say I want to find my helper T cells. I can go ahead and make sure that I'm choosing an AND gate as a logical kind of operator, and I can gate CD3 positive. And now I'm going to just highlight only those populations that are CD3 positive. And oh yeah, there's our T cell compartment on the far right of this tri-map plot. Now what about which of these islands are T4? Well, we kind of already know, but let's try gating here. And that's going to indicate that we're CD4 positive with this little kind of arrow showing that we're also CD4 positive. If we want to make sure we remove that tiny little island of double positives for the CD8, I can switch my logical operator to a NOT gate, gate that as well, and now we know that this is CD3 positive, CD8 negative, and CD4 positive. And that's the clusters that we have selected here, which should match up with those clusters that we were highlighting previously via the heat map, but a little bit more convenient way to do that. And then one other really cool thing about this plot is it comes with a legend. So if you right click this, copy to clipboard, and you paste this somewhere, kind of run out of real estate here, you can paste and you'll get not only your chord plot, but you've got this handy little legend here which tells you which color corresponds to which cluster. So now if you're going back to your workflow in FlowJo, you'll know exactly which cluster corresponds with which of those populations of interest based on your gating. Okay, and with that, I was hoping to get into a little bit more ViolinBox plots plugin to show you guys how that visualization works, but I think I've kind of run out of time for this webinar, so I just want to thank you guys all for joining. I think a lot of people stuck it out to the very end of the discussion here. Last call for questions. Any questions that came up along the way, I'll go ahead and say drop those into the Q&A box and I'll try and answer as many as I can. Also, I want to put it out there, we have a lot of great help available via email. So if you guys ever want to reach out to me, I'll put my email address right here. It's just taylor@bd.com. We also have a great set of support folks that can help you out if I'm not available, and that's going to be FlowJo@bd.com. If you have any questions about FlowJo, the FlowJo product. If you have any questions about SeqGeek analysis, maybe you're getting into single-cell RNA sequencing and you want to try that platform out, definitely reach out to SeqGeek@bd.com, and these guys can help you with any questions around that software as well. Question coming in on the R path. So I'll put that up here in a minute, but first let me just try and paste that in. Oh, that's not going to work. I guess it's going to depend on whether or not you're on Mac or Windows, right? If you want to know the R path. So for Windows, it's going to be simply, question about webinars as well. It's one thing at a time here. So the default R path for Windows is a little bit trickier than any other. So I'm going to just type that in here and paste it into the chat. It's going to be roughly something like C:\/ program files, because this is where you installed R, and then R \r version, which in this case probably 3.6.1 for most people. This is going a little bit off screen, that's okay. And then make sure you go to bin x64. Let me move that somewhere where we can all kind of see it, hopefully. There we go. And I'll just copy all of this and put that into the chat. So you can copy paste that. Just double check me on this path, but if you're in your preferences, you can always go to diagnostics, choose. And when you click choose here, it's going to take you to a file browser. If you just go up one layer from where it starts you, you'll probably already be in the program files. So when you go up with that little green arrow at the top of the browser, you can go to R, check your R version, just double click on that folder, open the bin folder in there, double click on that, open up the x64 option, select that, and that'll populate in here if that's easier than just copy pasting. Okay, other question was, where is the webinar going to be available after this? We do post all these webinars on our kind of legacy webinars page. If you go to FlowJo.com, I'll navigate there now. You'll see there's a section for Learn at the top. If you go to Learn and click on Webinars, under Explore Previously Recorded Webinars, that's where this webinar will be recorded. So if you ever want to go back and look at what we talked about, you can definitely check out this page, and it's got all the different webinars from long while back. So if you ever want to refresh, this is a really great place. You can kind of just, have your coffee, do some analysis, and listen to one of us yammer on about analysis in FlowJo or in SeatGeek. Sorry, I missed a question early on. The question is, what's the relationship between FlowJo and BD Help Support? So FlowJo is now part of BD. We're a subsidiary of BD and officially known as BD Life Science Informatics. So BD Life Science Informatics has its own support team, of which I actually run the team here. So if you have any technical questions about support in FlowJo or SeatGeek, that's typically going to get filtered to us. And again, the email addresses that you should reach out to for any of that kind of informatics support is going to be flowjo@bd.com or SeatGeek@bd.com. And I'm always open as well, so feel free to write to taylor@bd.com and I'll get back to you as soon as I possibly can. Okay. Great. I appreciate you guys all taking the time and sitting in with me on this webinar and demo. I hope to see you guys in the next one. And until then, have fun out there in your data. Thanks, guys.